Hi, my name is Hannah Illig, and I'm a biology major with a concentration in secondary education.
Hi, my name is Ashley Zosky, and I'm a biology major with a concentration in pre-medicine.
Hi, my name is Mackenzie Cree. I am a biology major with a concentration in pre-medicine and a chemistry minor.
Hi, my name is Sarah Moehring, and I'm a biology major with concentration in pre-medicine and a chemistry minor. And today we're bringing you, does water influence anthocyanin pigment production in fast plants? This is our experiment.
Brassica rapa, known as fast plants, their phenotype can be due to the environment or genes. This experiment was developed to figure out if the anthocyanin pigments, the purple pigments in fast plants, are due to genetics or environmental factors. Many plants belong to the Brassica rapa family. However, fast plants were developed to grow quickly for scientists to study the genetics of these plants. A study done on one of the family members of Brassica rapa, approximately 2.69 to 61.21% of phenotypic variation is due to pigment synthesis genes. However, they found no biosynthetic genes related to anthocyanin pigments. Anthocyanin pigments can be due to pH, light, structure, and temperature. Anthocyanin pigments can be predicted by test cross. This made it easy for us to see if the environment actually did have an influence on the pigments. We selected the F1 seeds. The F1 seeds were predicted to be 100% purple.
We hypothesized that the amount of water will affect the appearance of the anthocyanin pigmentation in the fast plants. Optimal water of 15 milliliters of water will show a high amount of pigment. 5 milliliters will show little anthocyanin pigment, and 25 milliliters will show intermediate amounts of anthocyanin pigment. Methods. 6 of the F1 generation seeds were grown in small planting pots. All plants were watered at the same time with distilled water. Pots were planted with different water measurements. The optimal amount, 15 milliliters, wet, 25 milliliters, and dry, 5 milliliters. The pots were placed on a windowsill, indoors, facing direct sunlight. Anthocyanin pigments were measured using a scale of 0 to 4. The figure below shows the scale of anthocyanin expression in each plant.
The results of our experiment were based on each experimental trial that we each ran with 6 plants. Trial 1, most anthocyanin pigment was observed in 25 milliliters of water, and there was a 100% seed survival rate. In trial 2, most anthocyanin pigment was observed in 25 milliliters of water, and there was a 67% seed survival rate. In trial 3, the most anthocyanin pigment observed was in 5 milliliters, with some being observed in 15 milliliters of water. There was an 83% seed survival. Lastly, in our 4th trial, the most anthocyanin pigment was observed in 25 milliliters of water, and there was an 83% seed survival. That means that overall, the average amount of anthocyanin pigment found in our fast plants was 0.571 in 5 milliliters of water, 0.214 in 15 milliliters of water, and 2 in 25 milliliters of water. Our data overall was inconsistent, as there was no specific water amount that was ideal for phenotypic expression of anthocyanin pigment. Also, the anthocyanin pigment we observed was at its highest when fast plants first sprouted. Then as they continued to grow, the pigmentation would fade. The last figure is the percentage of seeds of fast plants that grew in our experiment.
Overall, the results of our experiments varied with each experimental trial that was created. Some plants expressed the most anthocyanin pigment in 25 milliliter conditions, while other plants expressed the most pigment in 5 milliliter conditions. We used F1 generation seeds that were all expected to have a 4:0 ratio of purple plants. Since all of the plants that we grew were not purple, it can be concluded that phenotype was influenced by more than just genetics. that it was influenced by both genetics and the environment. These varied results also allowed us to draw a conclusion about the experimental conditions that we decided to use. Since the results were so inconsistent, it can be concluded that water alone does not influence the amount of anthocyanin pigment that the fast plants produce. We reject our hypothesis as the environment does play a significant role in the expression of phenotype. However, water alone is not one of the environmental factors that has a significant effect on the phenotype. Other environmental factors that could have influenced our results are the amount of sunlight that our plants received, as well as how far away they were kept from the windowsill. In the figure below, the circled 15 milliliter plant was the plant when it first started to sprout, and you can see that the purple pigment is seen. And as the plants continued to grow, the second picture, you can see that the purple pigment has faded away and they are green.
Thank you for coming to our presentation. I hope you enjoyed it.